Okay, hi everyone. Thank you for attending this presentation and your interest in our work. My name is Laila El Khattabi. I'm an associate professor at Sorbonne University Hospitals in Paris, France, and I'm going to present some novel and preliminary data for our prospective study CHROMAPS. So here are my disclosures. I don't have any conflict of interest to declare. So to start, just a little presentation of what is the CHROMAPS study. So it's a prospective clinical study to assess the medical performance of optical genome mapping and long-read sequencing for the detection of numerical and structural chromosomal aberrations. So this study has been funded actually by a French rare disease call, which aims at solving diagnostic deadlocks. So when we designed this study, this was in a context where we, as you all know, our current strategies to diagnose chromosomal aberrations present several limitations depending on the technique that you will be using, such as resolution for karyotyping, for example, an ability to detect or locate structural variants for DNA microarray, for example, and also an accessibility of highly repeated regions for genome sequencing, for example. So the current technological breakthroughs, in particular optical genome mapping, as well as long-read sequencing, offers the possibility of detecting additional chromosomal aberrations that we were not able to detect so far. And you all may have read this paper from Chesson et al. In 2019 that clearly shows that sequencing and optical genome mapping have different abilities to detect different structural aberrations, and they are clearly complementary to each other. So in this context of technological breakthrough, actually, we can expect a major change to come in clinical cytogenetics, but to allow this to happen, actually, we need some clinical validation. And so how these new technologies could benefit to the diagnosis of constitutional chromosomal aberrations is still an open question. And this is because we are lacking clinical prospective studies. So that's why we proposed this French multi-center prospective study, where 14 inclusion centers are involved. Most of them are in Paris, but not all, actually, and several ones are outside from Paris. And the main objective of our study is to compare the diagnosis rate for chromosomal aberration detection using optical genome mapping or long-read genome sequencing compared to standard of care tests. And our primary endpoint will be the percentage of class 3 to 5 chromosomal aberrations detected by OGM or long-read sequencing compared to the same percentage detected by standard of care tests. And some of our secondary objectives will be to estimate the incidence and distribution of chromosomal aberrations, to evaluate the limitations of OGM and long-read sequencing, as well as the impact on patients' disease management, and to estimate the medico-economic impact. So for these secondary objectives, we will focus mainly on the type of aberrations that will not be detected by OGM or long-read sequencing. So these will correspond to false negatives, as well as the percentage of patients for whom OGM and or long-read sequencing will detect clinically impactful unknown aberration using standard of care tests. Among other objectives that we have in this study is to evaluate some practical aspects in a clinical setting, such as the organization around the processing of the sample, the turnaround time, the robustness of the protocol, as well as the variant interpretation burden, which is a key point in a clinical setting. We also aim at optimizing the analysis workflow, as well as the variant selection pipeline, and also establish some recommendations for the analysis strategy. So for this, we are now setting a user group, French user group, that is discussing all these different aspects. We also aim at spreading the knowledge around the technologies and also share our experience. The sample size has been actually calculated by statistical analysis, and our inclusion of 300 patients over 24 months. And actually, we've been delayed by some administrative procedures, so we will be extending the duration by 8 additional months. So 2 groups of patients will be studied. The first one presents with reproductive disorders, and this one will be 200 patients who have at least a karyotyping as a standard of care analysis. And the second group corresponds to patients presenting with neurodevelopmental disorders and or multiple congenital aberrations, and this is 100 patient size group who will have as standard of care genetic analysis at least a DNA microarray, which is currently done systematically in France for this indication, as well as in some cases exome sequencing or even short-read genome sequencing. For these 2 patient groups, we will, in addition to the standard of care genetic analysis, perform OGM and long-read genome sequencing using nanopore as inclusion criteria. So we've defined isolated neurodevelopmental disorder or associated to MCA, as well as multiple congenital abnormalities with or without actually neurodevelopmental disorders. And for reproductive disorder patients, these concern females presenting with primary amenorrhea and or premature ovarian failure insufficiency before age of 40, as well as non-obstructive azoospermia or severe oligozoospermia below 10 million per milliliter for male reproductive disorders. And actually the last indication is currently not included, but we are in the process of including also recurrent miscarriage patients. So for neurodevelopmental disorders, we will not include cases in which there is a context of perinatal suffering, such as hypoxia during labor. And for male reproductive disorders, we are not including patients with obstructive azoospermia. In terms of organization, all inclusion centers will perform standard of care analysis on site, and actually samples for optical genome mapping will be sent to 3 different centers that have been equipped with the Sapphire. And for long read sequencing using nanopore, this technique will be led in 2 different centers, Lyon and Pitié-Salpêtrière. So afterwards, the analysis will be obviously performed in a blinded way, and results will be compared after anonymity lifting. So I'm gonna show some very preliminary results that I'm exclusively sharing with you for samples that have been included between September 25th on 2022 and December 21st on 2023. We have included 224 patients, 4 of which have been excluded for different exclusion criteria. And among the 220 retained patients, 118 presented with neurodevelopmental disorders and or multiple congenital abnormalities, and 102 presented with reproductive disorders, 26 of which had premature ovarian insufficiency, and 76 had azoospermia or oligozoospermia. Here is the representation of the distribution of standard of care techniques, and you can see that in half patients DNA microarray was performed alone, in 40% karyotyping was performed alone, and in a little bit more than 10% DNA microarray and karyotyping were performed. The diagnosis rate for karyotyping, so in reproductive disorders, was of 8%. And the diagnosis rate for DNA microarray in neurodevelopmental disorders was 15.6%. So just to put this in comparison with what was used to perform the statistical calculation of the sample size, we were expecting 5% of diagnosis rate for karyotyping and an improvement, a doubling of this diagnosis rate using OGM and or long-read sequencing to 10%. And this led to a calculation of a sample size that was 155. So you can see that we are already above our estimated percentages at the beginning. So we could even include less than 200 patients in reproductive disorder group, and this should be enough for statistical power. And we also hypothesized that DNA microarray would have a diagnosis rate of 15%, and we are actually around this same exact percentage. So on these 220 samples, 119 were already run for OGM. So I'm going to present only results for optical genome mapping, Bionano, actually because we still didn't start the long-read sequencing part of the study because of some, let's say, administrative difficulties. So among these 119 samples, 89 have now a completed analysis and 30 are ongoing. We've chosen the following settings. So CNV size above 500 kbs, SVs without real size threshold except for insertions, for which we are only selecting the ones that are above 5 kbs. We filter out SVs or CNVs which are present in more than 1% of the control population. And we are also having a look at CNVs which have genes at 500 kbs around. So among the 89 samples for which the analysis is completed, we have detected 16 aberrations. You can see here some of the ones that are classified as class 4 to 5, which means likely pathogenic to pathogenic. So these are 9 aberrations. Among which 2 were actually previously unknown from a standard of care analysis. You can see on this on the right side of the table the results from OGM. So the check means that the aberration that was previously detected either by karyotyping or DNA microarray was actually detected also by OGM. And you have 2 additional results that we will be exploring a little bit later. So here highlighting one specific aberration, which corresponds to 3 CNVs actually. So here you can see DNA microarray profile. So these are SNP microarrays. That's why you have 2 different profiles, one for the B allelic frequency and the other one for the log-R ratio. So you can see one small deletion on the short arm of chromosome 9, a bigger deletion in the long arm of chromosome 9, and a large deletion on the short arm of chromosome 9. So this patient has been explored by in situ hybridization, actually, with 2 different probes. The red probe has been placed on the duplicated segment, the large duplication that you've seen on the short arm of chromosome 9, and the green probe has been located in the large deletion on the long arm of chromosome 9. So in the in situ hybridization picture, you can see the normal chromosome 9 with the red and the green probe, actually. And to the left, at 9, you have the derivative chromosome 9, where you can see 2 red spots corresponding to the duplicated region. And you can see that the red duplication, actually, the duplicated segment has been inserted roughly at the location of the deletion in the green probe. So this, you can see it much more easily in one-in-all technology, which is the OGM. So in the panel, you have the Circos plot for all chromosomes. You can see that you have some connections, some CNVs on the chromosome 9. So if you zoom in to chromosome 9, you can see on the CNV track some deviation of the curve. The red deviations correspond to the 2 deleted segments, one in the short arm, the other one the long arm of chromosome 9, and the blue deviation corresponds to the duplicated segment. And if you observe the pink connecting lines, you can conclude to the insertion actually of the blue duplicated segment into the red deleted segment on the long arm of chromosome 9. So this is a little bit complicated actually when you get used to those kind of pictures, and you can conclude to some mechanistical situations because of some cytogenetic expertise. And here at least you have the zoom in into this region. You can see on the CNV track the red highlighted region which is deleted, the blue highlighted region which is duplicated. So this is on the short arm of chromosome 9. And when you look at the blue, the patient's map, the blue map, compared to the green reference maps, you can see that the duplicated segment actually is connected to a position on the long arm of chromosome 9, since you are connecting the position at almost 9.8 million base pairs is connected to a position around 74 million base pairs on chromosome 6. So yeah, just to conclude, in one technique, we've been able to confirm what was seen both by DNA microarray and FISH analysis. So if you go back to our table of 9 detected aberrations, here I've highlighted 2 aberrations that were not previously detected, and I'm going to present both of them. The first one is a deletion on chromosome X, actually. So this patient is presenting male reproductive disorder, oligozoospermia. And actually, OGM, the karyotyping was normal, and OGM has detected a small deletion STS gene region. So for those among you who are used to this region, you may know that this is associated to hypogonadism as well, which may be correlated to the phenotype of the patient, but STS deletions have also other dermatological manifestations. So we are currently in the process of collecting additional clinical data for this patient. And the second case, deletion of almost 2 megabases on the long arm of chromosome 5, which is NSD1 gene. So you know that this gene is involved in Sotos syndrome, which is a neurodevelopmental disorder, in addition to other associated malformations. And actually, we are still investigating this case. We need to confirm this aberration because it wasn't detected by other techniques. So it could be a false positive of OGM, but we are currently investigating this CNV. And in addition to these probably pathogenic or pathogenic CNVs, we have also detected 5 variants of unknown significance. All of them were missed by standard of care techniques. And here I'm showing one example concerning a patient presenting with a neurodevelopmental disorder. OGM has revealed an inversion that may disrupt SMARCB1 gene. So we need now to confirm again this SV and perform segregation analysis and explore whether this is inherited from a parent or not. And another case of variant of unknown significance is this small deletion in the first intron of FBXW7 gene, which actually encompasses very interesting regulatory elements. So just to put you in the picture, FBXW7 has been recently involved in neurodevelopmental disorders, and this is a screenshot from UCSC showing where the blue highlight shows the location of this small deletion, and you can see from gene enhancer, regulatory elements, and gene interactions that there are 2 different putative regulatory elements within this deleted region which interact with the promoter of FBXW7 gene. So here again, we have requested the parents' samples to analyze the inheritance of this SV. So in addition of those 14 variants of unknown significance or pathogenic variants that I've just presented, we have also detected 2 kind of pathogenic susceptibility CNVs, which are the known deletion 1q21.1.2. And so the other point that we've been evaluating is the interpretation time, actually. So we set a timer when we start the analysis, and we just evaluate the time that it takes to interpret each analysis. And surprisingly, this hasn't been that long. Actually, the median is 10 minutes, and the range is from 3 to 16 minutes per analysis. So to conclude on this very preliminary results, I must say that we had 2 major good surprises. The first one is the interpretation time, which I just discussed. And actually, this is still reducible since, to be clear, I've been interpreting all are referenced in DGV, for example. In some cases, the ones that contain non-coding genes or non-OMIM genes. So this could still be reduced if we filter out using DGV or OMIM genes, for example. And the second good surprise has been the technical improvements. So when we just started, there was this new protocol and the new version of chips, etc., which has simplified actually the technical part. The major observations coming out from this initial analysis is that all known aberrations actually have been detected so far. We know very well the limitations of OGM, which are centromeric breakpoints, etc. But for now, all aberrations have been detected, and as we've discussed in our paper from AGAG in 2021, in a clinical use, we would most probably associate OGM to some kind of limited chromosomal counts in GIMSA staining. We were also surprised by the amount of additional information that we've got. So potentially, again, this is very preliminary. We have some variants of unknown significance that need to be confirmed. Further investigations will be undergone. But for now, we have plus 70% of diagnosis rate in neurodevelopmental disorders and plus 60% in reproductive disorders, including, again, variants of unknown significance, but still, we have some additional information and variants to explore. And last, for now, we didn't have any failure for our samples. So there is more to come since we need to really refine the performance estimation for OGM, but also for long-read genome sequencing. We still need to perform this comparison between long-read sequencing and also OGM, not only in terms of diagnosis rate, but also in terms of feasibility, interpretation time, etc. And we even should be able to compare the results to short-read genome sequencing for some cases, since now in France, we have the possibility of performing this short-read genome sequencing in a clinical setting for patients presenting with neurodevelopmental disorders and or malformations. And also, we are excited about this ancillary study on recurrent miscarriage as well. So thanks for attending, and I have to acknowledge all my collaborators at APHP Sorbonne hospitals, but not only, also at Clermont-Ferrand, Lyon, Montpellier, Rouen, Cochin, Reims, Strasbourg, Montpellier, Marseille. Also my wonderful collaborators at Radboud, Nijmegen, and also University of Oulu now, Finland, so Alex Oshin, Cornelia Nivlin, and Tuomo Mantere, as well as our funders and the Bionano team for their support. So thank you.